In this video, I just want to clear up a couple of topics I missed out on in early videos. Specifically, we're going to look at two specialized cells that I missed out on, and we're also going to look at some of the industrial uses of enzymes. To start with, we're going to have a look at red blood cells. Their function is for binding onto oxygen that they'll pick up at the lungs and distributing it throughout the body through the blood. To do this, they have a few adaptations. For one thing, they don't have a nucleus, because the only job of the red blood cell is to bind oxygen on. It doesn't need a nucleus, and it would only take up space that could be used for binding on more oxygen. They contain a chemical called hemoglobin, which is used to bind onto the oxygen, and they have a special biconcave shape to increase the surface area available for oxygen to bind onto. Now that biconcave shape, if the red blood cell was just shaped like kind of a minstrel or something like that, it would have some surface area on which you could attach oxygen. But if you squish it in the middle, you'll notice that you've got a lot of extra surface area that you didn't have before. This concave shape, called so because it goes in like a cave, increases the surface area available for the red blood cells to bind onto oxygen. The other type of specialized cell we're going to have a look at are the ciliated epithelial cells that line the tubes in your lungs. So when you breathe in, air will go down your esophagus, your windpipe. It'll branch off on the bronchi and go into little tubes that will end up in the alveoli of your lungs. This kind of branching network again, like we saw with the circulatory system. The lungs are encased in a kind of membrane called the pleural membrane. You don't really need to know this, but just for your own information. And there's a muscle called the diaphragm at the bottom, which helps you to breathe in and out. Now, the bit I'm really looking at today is kind of the esophagus and the bronchi. We're going to look at how the lining of these structures helps protect you from infection. Because the problem is the air around you and the dust inside that air and all the little particles floating around are covered in pathogens, microorganisms like bacteria and fungi that would like nothing better than to get into your lungs to live. Because for them it'd be a great place to be. It's warm, it's moist, it's dark. It's a great place to be if you can get in and get past the defences. So your esophagus or your windpipe is where the air starts to go from your mouth down towards your lungs. If a pathogen like this bacteria here, doesn't really have a smiley face, was to get down your windpipe and into your lungs, they'd run rampant. That's why a lung infection is a pretty serious matter, because it's quite hard to get rid of them. Luckily, your body has some defences set up. In particular, the lining of the esophagus is covered with epithelial cells. Sorry, the label got a bit cut off there. Epithelial cells line organs, but these ones are a little special. Because as it is, the pathogens wouldn't really be bothered. Whatever, what are you going to do? But these epithelial cells have cilia, which is kind of like little hairs that stick out into the lining of the throat. Now alone they wouldn't do much, and the bacteria wouldn't be that bothered. But they work in combination with goblet cells. These are cells that produce mucus. Now again, that mucus is going to be used for something. In fact, I'll show you now. The mucus lines the lining of the throat. The pathogens get caught and stuck in that mucus, and then the cilia, the hairs, kind of wave upwards like a long Mexican wave, passing the mucus with the trapped pathogen upwards towards the mouth. Oh no. Okay, we're going to have a look at one last bit, which is two uses of enzymes that I didn't mention earlier on. One kind of obvious one is you can use it in biological detergents for washing your clothes. You can put digestive enzymes in that will break down stains. Now remember, the digestive enzymes, an enzyme isn't alive, so you can easily put it in, and as long as the temperature stays below about 40 degrees, they'll work really well. If you put it at a really high temperature, of course, the enzymes will get denatured. Again, have a look at that enzyme video if you need a recap on that. Okay, another use of enzymes is as an isomerase. Now we're going to look at this kind of bit by bit. But here we've got the glucose molecule we looked at earlier on, C6H12O6, and it's in a kind of hexagon shape. Fructose is another simple sugar. The only difference is this one, well, not the only difference, it's arranged into kind of a pentagon shape. But it's got the same formula. It's still got 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms. They're just rearranged differently from the glucose molecule. Because they've got the same atoms but arranged differently, they're called an isomer of one another. Iso means the same. You've just rearranged the bits. Just like if you got a Lego kit, you could rearrange it to make something else. You wouldn't be adding anything, but you'd be using what you have to make something else. Now we can use enzymes that will take that glucose molecule, that hexagonal glucose molecule, and change it into a fructose one, just by rearranging the atoms inside the sugar molecule. So first the enzyme binds onto it at the active site, and then we'll change it by rearranging the atoms into fructose. Again, the chemical formula hasn't changed. We haven't added anything to it. We've just moved the atoms around a bit. And the enzyme again is free to go on and do that to another substrate. So because this enzyme changes one isomer into another, from glucose into fructose, we call it an isomerase. Again, it's named after the substrate with an ase on the end of it. Okay, but why would we want to? Why would we bother using these isomerases to change glucose into fructose? Well, the answer lies in dieting. Fructose tastes a lot sweeter than glucose. So even though they both have the same amount of energy in because they've got the same atoms, you need to put less fructose on your food to make it taste as sweet as you would use glucose. So in other words, if you wanted to make your cornflakes tasty with glucose, you need a lot of it. With fructose, because it's sweeter, you wouldn't need as much. So if you use less sugar, you'd use less calories, and then you could lose weight.